share with you. One is the comments are so absolutely helpful, so I really appreciate everybody that's turned in a survey so far. Something that I've decided to do is I'm building a study guide for the next exam. I thought I'd have it done before I came in here, but I didn't. It's close, so it'll post later this afternoon. Okay. I have an idea for some extra credit activities. I'm just gonna I'll write it out so it's a little clearer, but just share what's rolling around in my head. The activity that I'm thinking about is that where you will find some papers, and I'm gonna give you a list of the topics that were on exam 1, and I wanted those papers to be dedicated to that topic. So you can do up to 2 of these, and you can do there's a couple of different things you can do for the activity. So like, one is you can record yourself, like make a PowerPoint and record yourself talking about that paper, and turn that in as a movie file. The other one is, this just kind of manifested itself in my other class, but you can develop a podcast where you are sitting there and talking to a friend about the paper. The other thing is just if you don't like, being in the spotlight like that, you can just write a paper review, which is like a 1-paragraph summary and then a 1-paragraph review of the paper. Okay? So the details of that will come out by Monday, and we will talk more about it on Monday too. All right? Questions? Suggestions? I gotta tell you, I don't know how to guide you through the podcast thing. I just know, I had some students do this in my other class, and it's turned out to be a really cool thing. But so figuring out how to build that is kind of on you, because I'm getting too old to figure these things out. Okay, and then as far as our Transcriptapalooza goes, there were only 3 presentations really left, and they were on Pull 3 polymerases. I'm gonna let those 3 groups off the hook, and I'm just gonna get those things posted so you don't have to present today. And I'm gonna try to finish up chapter 8, and let's get into chapter 10. All right. I wish I had the study guide for you so you using it right now as we're talking, but I'll get that posted later today. All right. So my thinking on that is helping you study for the next one. Anybody that really wants to improve for that next one, and then the points, the presentation, and just give me some points back if you weren't happy with exam 1. So those are my ideas. Thanks to your comments. Don't forget, we've got a quiz that is due on Monday. It's 6 to 9, so I'll try to get that study guide so you can hop through and see what you're comfortable with. Alright, I'm going to go ahead and change my display here. Okay, I think we ended up on Wednesday, we were talking about processing, regulating gene expression at the transcription level with microRNA. And now what we're going to finish up talking about is, we can regulate our RNA and all that, but let's back up and say, how does RNA what are the details about the processing of the mRNA? So remember, there's other RNAs, right? There's tRNAs, those other things that aren't ever going to be a protein, and so this stuff that we're talking about doesn't necessarily apply to those things. But the pre-RNA, there is a pre-mRNA. Remember that eukaryotes have the introns and the exons. There's also some other modifications that happen to those things. So they're pretty extensively modified before they leave the nucleus. The transcription, so opening the gene and getting manipulating the polymerase and getting the gene, the RNA transcript and the processing are kind of happening at the same time. So they're coupled together. So you guys now have this like really good picture of all of these darn transcription factors that bind and the complexity of all those things that bind the polymerase 2 especially. So now you can imagine that like we have that happening, and then we have the elongation of this transcript, and then even maybe before we're even done, there's some processing going on, or it's all happening in the same space. Okay, so the C-terminal domain of the RNA polymerase does play a key role to help bind some of the enzymes that are going to be processing or removing those introns out of our mRNA. So that C-terminal domain again, it's kind of fascinating, right? It has a lot of roles to it. Okay, so this is a nice flow, just kind of giving you a really zoomed out picture of what we're talking about with the changes that happen. So this up here is the DNA. That's the information that codes for the RNA. So this is what gets opened up and the polymerase binds and all that stuff. And here's what the pre-mRNA looks like. It is a complementary copy to the template strand, and then it also has, instead of having the T's in it, it's going to have the U's in it now, right? The light blue is the intron, and then the dark blues are the exons. Okay. So the first thing that happens is we have a 5' cap that's put on. So right up here, so the 5', remember the direction of our transcripts is 5' to 3', 5' carbon, 3' carbon. So we get a 5' 7-methyl guanosine cap that's put on there. The other thing that happens is we have a 3' polydenylation. So there's actually a cut. Notice this is shorter. There's a cut in the transcript, and then we end up adding A's to the end of it. And then after that, then we have the splicing. So we cut the intron out, put it together, and now we've got our protein coding sequence completely intact, and we've got some fancy little ends on the mRNA to, 1, protect it, and 2, guide it to where it's going. Okay, so this is what the 7-methyl guanosine cap looks like. There's a couple of really interesting things to notice in the picture. So here is our 5' end of our pre-RNA. Okay, and we've got our ribose sugar, our base, and the sugar phosphate backbone of it. And this is the 7-methyl guanosine that's added. So what's special about this is we have this methyl functional group on the 7th position of the nitrogenous base. So, and which is a different thing. And then we have this, the other thing that's interesting, instead of having like a 5' to 3' connection here, we actually have a 5' to 5' connection here. So it really is like a very, like if you think about like 3-dimensionally, it really is a different structure on the beginning of your transcript. So it's got a different shape, a different makeup to it. So it will protect from degradation because like the degradation enzymes, if you can imagine, this is the thing that they recognize, and that's the bond that they're going to break. But this bond looks completely different, right? So and usually RNases and things like that, they're going to chew one base at a time. And if they can't get the front one off, then it's protecting it. The other thing is it could be helpful in guiding it to the ribosome, along with other elements, the internal ribosome entry sites and stuff like that. Okay, so here is the poly A tail. Here is the DNA, just because it's always important to remember that where the information comes from, right? That it's coded in the DNA. So here is the transcript, and there's a couple of nice consensus sequences that are pointed out here. So you've got, there are some A-rich regions and some GU-rich regions, calling out the the RNA when we're talking about the U. But these are consensus sequences that are recognized by the polymerase itself, right? So as it's slipping along and it's building this stuff, it can recognize, oh, this is the slippery A region. Why do I say slippery? Because remember the A's and T's are like a 2 bond, hydrogen bond, right? So it's something that can be recognized. So, all right, so we have transcription, we build this long transcript, and then there is a key splice site. So once this is fully transcribed, then this splice site can be recognized, and there is a cut that happens after that CA. And then, that's an endonuclease cleavage. And then there's a poly-A polymerase, which is another polymerase, another enzyme, that will bind onto this end, and it's like, kind of works like no, it's not a reverse transcript. I'm mixing this up with telomerase, but no, this is a poly-A polymerase that will synthesize or put on adenosines at the end of this. So it just kind of pulls them in and puts them on there. So look at how long that can be. 200 of them can be on the end. So just because we have the picture up here, I don't want to scatter your brain, but if you were to pull all of the RNA out of the cell and then ask yourself, like, I really want to look at what RNA is here, but RNA actually does degrade really fast because we are dripping with an enzyme that is there to destroy RNA. So it's very transient. Doesn't exist that long. But if you really wanted to have a chance to look at it and you want to look at all of the RNA in the cell, you can convert it to cDNA. And so what is the primer that you can use to convert every single RNA in your, mRNA in your cell? What primer could you use for your cDNA synthesis? We always use an oligo-DT. So it's like 16 T's, it binds right here, and then we can use this as the template to convert it to DNA that lasts for, years. So that's how that works. Okay, so we've looked at the 5' 7-methylguanosine cap and then the poly-A tail. So now the next thing is, how does the intron get pulled out? So this is a zoomed out look without adding in all of we're just looking at the DNA elements, the RNA elements. And then in the next slide, we're going to look at the proteins and the RNAs, the other RNAs that work to do this. Okay? So what happens with the mRNA is there is a 5' splice site. So there are more consensus sequences within the transcript. So sequences that can be recognized, right? So we've got a GU on the 5' splice site. We've got an AG on the 3' splice site. And then somewhere in the middle position, 2/3 of the way over from the 5' is a branch point A. Okay, so it's a key adenosine that's right there. All right, so first thing that happens is we have the cut at the 5' splice site, and the G actually rolls over and it will interact intramolecularly with that A. So the G and A come together and branch in a branched interaction. Then we have cutting of the AG on this side, the 3' splice site, and simultaneously we have then the exons pulled together and ligated together. So a lot of chemical reactions in here. We have cutting, we have ligating, we have rolling that whole strand so that it can make an interaction and all that stuff. So this is the more complicated, detailed picture showing you how these things will work. So there are a number of small nuclear RNAs, which is what the SNR is. So small nuclear RNAs. And they are actually RNA molecules. So down here, look at this first. We have the U1 SNRNA, and you can see it kind of looks like a tRNA, but it has a it has a complementary sequence here on this little arm to that 5' splice site. So the GU and everything, and what's around the GU, it can bind to it. This thing is actually built into a complex of other, like up to 10 proteins are like around it, carrying it to do what it needs to do. Okay. Okay. So when you're looking at what we're calling the spliceosome, which is all of these nice, you know, all these little players coming in here, you have to picture that this is RNA that is folded up in these globular 10 proteins are around it, like holding the RNA. So it's RNA and protein is the spliceosome. Okay, so let's walk through the process. We have the U1 SNRNP. That's what we call it an RNP. So it's a small nuclear ribonucleoprotein is what it is called. Did I say that right? That's such a mouthful. Ribonucleoprotein particles. Okay, so the U1 binds that 5' splice site, you know, recognizing that the RNA sequence by complementary base pairing, and then that binding starts that bending, so giving us the right shape of the RNA transcript. Then the U2 comes in and it binds it gets into the position where that the branch point adenosine is at. So that's really important. That's what the U2 does. And then we bring in U4, U6, U5. All of these things come in together and they catalyze the reaction to start basically pulling the G from the 5' splice site to the branch point A. Okay? So we have a couple of movements in here that I haven't mentioned. So we end up U1 and U4 eventually do fall off. And we're left with U5 and U6 alone with U2. And we have this, once we have this conformation of the spliceosome, then we have the formation of what's called the lariat-like intermediate. So lariat refers to that rolling of the RNA and then connecting it to itself. So we have that, we bring it into that shape, and then you have excision of the introns and ligation of the exons. So the cutting and the movement, all of that is happening by this machinery here, and it kind of happens at the same time. You know, so it's like you know, you're building a recipe, right? And you're cooking, and you're like throwing in the onions and the garlic and everything at the same time. It's like that. It's like all of the stuff is happening, you know, in space and time at the same time. So we end up with the lariat intron, and then we end up with that. And something I didn't mention, but you know, we're processing this RNA, and we're cutting a lot of stuff. So where does that go? It just gets degraded. You know, the other that's free to be degraded by the RNase or specific nucleotide degrading machinery. I skipped over all these. Okay. So that's how the SNRNPs do it. There are some self-splicing mechanisms also. So some RNAs will self-splice also. And it's the same, you know, same thing that we're talking about. You know, the steps don't really change, but sometimes the RNA can almost do its own attack by itself. Does that make sense without the rolling of it? So self-splicing is when the RNA itself catalyzes the removal of its own intron in the absence of those SNRNPs. Okay. So there's two groups of self-splicing introns. We have group 1s, and that is over here. And group 1s begin by cleaving the 5' splice site mediated by the guanosine cofactor. So it's like there's a guanosine cofactor that attacks the 5' splice site. So just a nucleotide coming in. All right, the group 2s are, there's cleavage of the 5' splice site. It results from attack by an adenosine nucleotide in the intron. So the adenosine itself is the attacker in the group 2s that causes the cut and that. That's a little harder to picture. I don't know, everybody could be different, but it's like easy to picture the machinery doing it, and it's a little harder to picture the RNA doing it by itself, but it is possible. At this level, what we're looking at is the nucleotides themselves within the RNA are doing the chemical attack, the organic chemistry in that, right? Okay. So we've got all this processing of the RNA, but there's and you've got like alternative splicing we talked about, right? So you can get from single transcript, you can get these different RNAs. But then there's even, in addition to that, there's even RNA editing. So we can make a transcript, we can get it all put together so that it has the coding sequence, but there's still editing at, we can still change a single nucleotide. So this RNA editing is where we, it involves just a single base modification. And there are like chemical functional groups that can be added and things like that to change it. So deamination, just in a couple of examples, you can have deamination of cytosine to give you something that looks like uridine, and you can also have deamination of adenosine. It gives you something called inosine. So you get these different nitrogenous bases by doing a chemical reaction on those, changing the functional groups on those ring structures. So here in the diagram we have the apoproteins, right? And in the this is the transcript. If it's unedited, then which is the case for the liver, we have, when it's translated, we get ApoB100 protein that has its job. And then you have, but in the intestine, there's an editing event, and we have the C that is deaminated to a U, and we end up with UAA, which changes a coding sequence. We're talking about in chapter 10, and we get apoprotein B48. So probably a lot of similar domains, but maybe the active site's different, or whatever it's able to find is going to be different, and stuff like that, right? It's interesting, you can change a single nucleotide, and then you'll change a single amino acid, and then you'll get this completely, well, not completely, but you'll get a different function in the protein. Okay, so mRNA degradation. Kind of mentioned it a few times, but degradation is typically initiated at the poly A tail. Okay, there's 200 of them, right? So that's, quite a lot. So there, it's like sometimes I think of this as like there's a race to get the protein, right? For those 200 nucleotides to be pulled off before you don't have the information anymore, right? So rapid degradation of mRNAs contain specific AU-rich sequences. The rapidly degraded mRNAs have a lot of AU-rich sequences near the 3' end. But the enzymes that go to work are the nucleases. So here is our mRNA with the poly A tail. We have deadenylation. So that's when the poly A tail is gone, and we just have chewing off of each nucleotide by a nuclease. What does it do? It's like a hydrolysis reaction that breaks that the sugar phosphate bond, the covalent bond, right? And then you can have also decapping. So that just because we're calling out the poly A tail doesn't mean the 5' end doesn't have anything happening, but there are enzymes that will decap the 5' end, and then nucleases will chew that A nuclease is an enzyme. When you think about this type of enzyme, it's different from a restriction endonuclease, where they're very specific and they recognize a specific sequence. Nucleases just chew nucleotides, or nucleic acids, those chains. They just break them. They don't care what the nucleotide base is, they just break that bond. And it's one at a time. I love that they give it like a Pac-Man, view, so it's like it's chewing it, because that's kind of what it does. It chews off the end. Why is that important? I should say, why is it so important that they're degraded? Because we need proteins when we need them, and we don't need them when we don't need them, right? So you don't need the RNA continuously doing that, which is the whole reason why the new, RNA-based medicines are interesting, is the idea of them being transient, short-lived. Okay, so hopefully by now you've got this really detailed picture of transcription, right? We know you can sit together, if you can get together in your groups, start off with okay, let's open up let's talk about the promoter, talk about all of those DNA elements, and then opening it up, and what does it look like? Be able to rattle off what it looks like for a POL2 gene, right? What are those darn TF2s and stuff, right? And then just be able to rattle off basically the life cycle of a transcript, right? So how does it begin? How does it get processed? How does it end for it? All right, we're almost at 10:30. It's almost over. So we'll start with translation. So we can move on. Don't forget our quiz is just chapters 6 through 9. So this isn't on the quiz for this week. Translation is now we're going to go following gene expression. We're going to take our mRNA and we're going to have it built into a protein. This is a really, really detailed picture of a ribosome on its side. We've got the small and the big part of it. So it's a really cool thing because you see a lot going on here because we're seeing the amino acid sequence, but remember there's also RNA folded in here as well. Okay, so translation is that second step, and it is another level of gene expression, and it's another level where gene expression can be regulated. Turn it on, turn it off here as well. The multi-step nature of molecular biology is actually a really cool thing because of all this chance to make it right. Okay, so here is our central dogma. So we've got our translation. I always remind people that this is where we're changing the language. That's why the term there, translation. So we're going from a nucleic acid to an amino acid. Pretty different structure. There is going to be an adapter involved. So this green thing that we see in the picture, that's the tRNA. And it's got a job where it reads 3 nucleotides at a time. And those are connected or associated with a very specific amino acid. So that's how the reading and the translating goes through the adapter. All right, so they're synthesized. Our proteins the information for the protein is in the mRNA, and the process for translation is very conserved. You're going to see very similar things happening in the prokaryotes and the eukaryotes. The ribosome gene and the protein might be different, like the sequences, but their overall structure and shape is the same between prokaryotes and eukaryotes. And then how they grab on to the RNA and how they read the codons is pretty conserved, all that stuff. So there's direction that's important. The mRNAs are read from the 5' to the 3' end. Okay, and the polypeptide chains are going to be synthesized from the amino to the carboxy terminus. So remember the amino acid has that central carbon and it has an amino side and a carboxyl side. And so as we're reading 5' to 3', these are organized in that orientation. It's it's the N amino terminus to the carboxyl terminus. You'll also see it said it's the N terminus to the C terminus. Okay. So, because what's the big deal with, why is it N? Because what's an amino? It's the NH group, right? Okay, so each of the amino acids is coded for by 3 bases. So this is where we start talking about reading frame, right? So we got to be in the right frame of 3. So 3, 3, 3, 3. That's how we're going to be reading that. The big worker, the player here is going to be our ribosomes, and then we've got our tRNAs as those adapters. So here's your genetic code. Anytime you see that you're going to be quizzed on, telling me, okay, what protein's going to come out of this mRNA, you're going to get a table. You do not need to memorize the code. I get that question all the time. You don't need to memorize that. Okay, you do need to know how to use it. Learned it for earlier classes. But it's a nice little table setup. So you've got the U. You start off with your first letter is on this side, and then your second letter is there, and then your third letter is there. You go find your 3-letter codes, and then next to it in brackets sometimes shows you what is that amino acid going to be for that one. It's a redundant code because some 3-letter codes will code for the same amino acid and all of that. What else can we review about this? So we've got our non-coding or nonsense codons. There's no tRNA that carries an amino acid to that, right? So there's a halt, and that's the stop. Every protein begins with a methionine because the start codon is AUG, and the amino acid brought into that is methionine. So when you become like if you're a genetic engineer and you're sitting there looking at sequences all day, which can make you go blind, you actually you will kind of get pretty good at being like, oh, okay, I see. You get like a sequence, the FASTA sequence, and you're like, oh, there's the start codon, and you might get as good as being like, oh, here's the end, the UAA or the UAG, something like that. Okay, so the tRNA, so the tRNA looks like this. We can, in panel A, we get this really nice simplified, flattened view of it. It is an RNA, so we see that they have a 5' end, and we have a 3' end. So the key features of this are, that we have these intramolecular hydrogen bonds. They're complementary nucleotide or bases to each other to give us these like 3 hairpin loops in there. This is given such a shape that we have 3 tRNA nucleotides that are in position to read the coding sequence, and these are called the anticodon. The anticodon binds the code, the 3-letter code. Okay, all the way on the other end is where we have our amino acid attachment site. So they're really cool. I think we'll talk about this a little more in the next slide, but there is the addition of the amino acid here. So something has to read that and then attach the correct amino acid to it. So this is looking at the more of the secondary structure. So it does get like a helical shape to it, and this is a nice space filling model, just reminding you that this is a 3-dimensional thing, right? It's a 3-dimensional thing. Okay, so how do we read how in the world do we get the correct amino acid attached to the correct tRNA with that anticodon? And it is the amino acyl tRNA synthetase. So it's an enzyme to make the whole world go round, right, in the cell. So this is the enzyme that does it. It's the amino acyl tRNA synthetase. And so what this does is it is an ATP dependent reaction. Why? Because I'm teaching this in biology 1, so I'm going to make you remember this. But We're taking things that are simple and spread out in elevated entropy state, and we're bringing things together and we're decreasing the entropy. So it's energy dependent. It's an energy dependent reaction. So we have, so here we have this is our amino acid. So you got that central carbon, the N terminus, and the C terminus. The R is the functional group, just a reminder, whatever it is, basic, acidic, whatever that thing is. And so we have the addition of an AMP, so we break ATP, which is a really energetically favorable reaction in the cell, right? We cut ATP, we get all this energy from it. We actually cut off the 2, and we apply the AMP to the amino acid, so we get an amino acyl AMP. So essentially, we basically we transferred the energy to the amino acid by putting that amino acyl AMP on it. Now, the amino acyl transferase will bind our tRNA, and it actually will read the codon. So we don't have this pictured here, but if you imagine this enzyme has an active site where there's going to be a very key interaction with that part of it, okay? And then it's going to be positioned to pull in the correct amino acid. So it reads and recognizes. Okay. All right, so then so that enzyme catalyzes the reaction where we pull the AMP off of the amino acyl AMP, and then that amino acyl group is put onto the tRNA. So now this whole molecule right here is called the amino acyl tRNA. That word is going to drive you crazy, amino acyl. But that's any questions about that mechanism? So when somebody asks you, how does the amino acid get onto the right tRNA, and hopefully you have a picture of that. So there are 20 enzymes that recognize single amino acids and the correct tRNA. Okay, there are 20 amino acyl tRNA synthetases, 20 different ones that recognize the different codons and then pull in the correct amino acid. All right, so now the decoding stuff. So the tRNA is our adapter. It's the thing that decodes the mRNA codon, and it complementary base pairs with the coding sequence. Okay, this is interesting. Some tRNAs do have the ability to recognize more than one mRNA codon because they do something called wobble base pairing. So, okay, so here we have a phenylalanine tRNA base pairing with UUC. Okay, so UUC is able to hydrogen bond with that anticodon. Okay, but we can also get, so we know this, right? We know these hydrogen bonds for a G and a C. That's how it works. But